This is the narration for the nervous system chapter, chapter 38 for biology 2. So you guys need to know that nerve cells are neurons. So neurons are nerve cells. It's the very first bullet. And then we're going to talk a little bit about later on which part of the nerve cell specifically receives, processes, and transmits information. The glial cells assist in the function of the neuron. They provide nutrients. Remember, nutrients are food for the neuron. They regulate the extracellular fluid in the brain and the spinal cord. Extracellular fluid is fluid outside the cell. And then the big thing on this first half of this chapter test is understanding how neurons communicate with each other. So the functions, the big things I had you guys underline: neurons receive information, they process that information, they conduct a signal to another neuron cell, and then they transmit the information. So this is a big thing on your test. You need to know the parts of a neuron. Like I told you guys, there's a picture in your textbook of a neuron. You need to be able to label that, and you need to understand what is going on in each part. So what's it basically? So dendrites are similar to branches. They're the branched part of the neuron. They branch off and away from the cell body. Their function is to receive information from the inside, internal, and external environment. When we talk about surface area, when you increase surface area, you increase the ability to receive the information. And then we talked about the senses. There's 5 senses: the ability to see, hear, taste, smell, and touch. So the sensory neurons are really important, and they have special adaptations that let them receive signals and respond to signals from the external environment, because your senses are external. And then lastly, on this slide, neurotransmitters. Neurotransmitters are messages received by the dendrites and neurons in the brain and the spinal cord, and they respond to chemicals. So these neurotransmitters, simply put, are chemical messengers that send messages from one neuron to another neuron. The cell body is the base of the neuron. The main function here is it processes information. It processes information in the form, it sends out information in the form of action potential. An action potential is an electrical signal made by the neuron. So it's kind of processed and made in the cell body. The cell body does contain organelles. So the cell body is where you're going to have metabolic activities occurring. That means metabolism, remember, is the process and making of energy. So the axon is really where the action happens. They're long and thin. They extend out from the cell body. Their function is to conduct action potentials from the cell body to the synaptic terminal. So the synaptic terminal is a meeting place from one neuron to another. So the synaptic terminal is where the axon of one neuron meets the dendrite of the next neuron. And we talked about pre and post synaptic terminals. The pre is the first neuron, the post is the second. Neurons are the longest cell in your body. Axons can stretch from your spinal cord to your toes, so make sure you know which is the longest cell in the body. And make sure that you know that nerves are bundled up axons. So axons are bundled together into nerves. So the synapse, where the neurons communicate. You have your synaptic terminal, you have your dendrite. So what I just said, you have the axon of one neuron meeting the dendrite of the next, and then there's a little gap that separates these. The output of the first cell becomes the input to the second. That means the message going down the axon of the first is going to be sent to the next neuron to keep the message going until it gets to the right recipient. All right, so figure 735 on page, figure 38-1, page 735. I'm going to get my book so you can follow along with me. And that's figure 38-1, page 735. We went through this in class, but make sure you know the synaptic terminal. Make sure you can label that. Make sure you can label dendrites and cell bodies. Make sure you know that the action potential travels down the axon. So just be able to label that main part, the little part on the left where you see the little circle. Don't worry about that right now. So neurons make and transmit information. Information is transmitted through electrical signals and neurotransmitters from one neuron. And then we talked a lot about resting potential and threshold and action potential. Resting potential is when the outside of the cell is negative. So we have a negative voltage. You guys need to know negative 40 to negative 90 millivolts is considered resting potential. When the neuron is stimulated, you're going to become more positive. Once you get enough positive charge, you're going to reach threshold. And when you reach threshold, that have an action potential occurring. So the voltage goes up super fast when you're going to threshold, and you get to plus 50 millivolts inside the cell. Action potential doesn't last for very long, and once action potential occurs, you go right back to resting. So the plasma membranes of axons are specialized to conduct action potentials from a neuron cell body to the synaptic terminal. So myelin is just something that helps the message move faster, the action potential move faster. It's this type of insulation that speeds up conduction, and it really allows the signal to jump from one axon to another axon. So it helps the signal go much. Now, what we talked about in class, and you guys read and summarized this, it's on page 738 through 739. This is a short answer on your test. So I have your test in front of me, and you have 25 multiple choice on your test, and then the rest is going to be in the form of short answer discussion. So when we talked about in class on page 738, one of your short answers is going to be potassium permeability produces resting potential. Discuss how that happens. So you know permeability means the readiness or the capability of the plasma membrane to let certain things in and out. So we talked about the sodium-potassium pump, but in this question, you need to talk about potassium. So how does potassium allow for resting potential? Potassium concentration is greatest inside the cell. Potassium has a positive charge, remember. So what happens is you have what's called resting channels, resting potassium channels. And when there's too much potassium inside the cell, the potassium will start moving outside through those resting channels, and it keeps moving out until you have that balance and you can maintain the right charge for the membrane. So simply put, potassium moves into and out of the cell through the resting channels until you get that negative charge reached. And that's how potassium maintains resting potential in the cell. Now the next short answer you'll have is changes in permeability to sodium and potassium produce action potential. So in this case, sodium is going to flood inside the cell. You have sodium channels as well, and during the action potential, those sodium channels will open, so the sodium will rush in, change the charge from negative to positive, and that's what creates the action potential. As soon as that action potential is sit down the axon, those gates will close, and the potassium gates will open, and potassium will flood out to create that balance, so you go back to resting potential. So it's always just trying to keep that right balance. Either you're going to create an action potential, or you're going to create a resting potential. Now the last you're going to have, action potentials are conducted down axons without changing amplitude. Discuss how this happens. So in your book, they tell you action potentials are all or none. If you don't reach threshold, you're not going to have an action potential. It's not until you reach threshold that you're going to have that action potential occur. Action potentials start where the exon calms away from the cell body. Because remember, the cell body processes the information and then sends out the signal, and the axon is where the signal is sent. So as sodium enters the axon, its positive charge repels other positively charged ions in the axon cytoplasm. When sodium enters during action potential, its positive charge repels other positive charged ions further along the axon. So almost instantaneously, the membrane potential of nearby areas to become more positive, and exceeds threshold. That causes sodium channels to open up and starts a new action potential. Sodium enters at these locations slightly farther along the axon. That starts the whole process over. Because a new full-size action potential is produced again and again all the way down the axon, the action potential travels to the end of the axon without losing voltage. As the positive charges pass a given point along the axon, the resting potential is restored as potassium channels open and potassium flows out. Only a tiny fraction of total potassium and sodium in and around a neuron is exchanged during each action potential, so the concentration gradients do not change very much. In the long run, the activity of sodium-potassium pump maintains the concentration gradient on both ions. So the question was, action potentials are conducted down axons without changing amplitude. Discuss how that happens. So basically, sodium is flowing in and potassium is flowing out all the way down that axon. Sodium flows in, creates an action potential. Potassium immediately flows out, restoring it to resting. Remember, the axon's very long, so you've got to keep that sodium, potassium flowing in all the way down the axon to make sure that the action potential makes it all the way to the end. So all the way down, sodium's flowing in, potassium's flowing out. So once the signal has gone through a certain part of the axon, that part of the axon goes back to resting. Then you have the next part of the axon going toward action. Then it goes back to resting. So all the way down the axon, you have the sodium-potassium pump working to maintain action potential and resting potential. So that's 738 through 739. They have, on page 739, more information about the flow, as well as on another slide, I have a couple of animations, and you guys can search those animations on YouTube yourself to find some animations for how action potentials work. So just a definition of action potential is a packet of information that goes down an axon. Once you reach the terminal, the info has to be transmitted to another neuron or to a cell in a muscle or a gland. And then that electrical synapse occurs, and I give the example in your textbook, within the heart. So with electrical synapses, you have signals can pass from neuron to neuron through gap junctions that connect cells, and a good example of that is the heart. But most neurons actually use chemical. So chemical synapses transmit information from one neuron to the next. The neurons don't touch the synapse. Remember, there's a little gap, and that's called the synaptic cleft. And we've talked about the presynaptic cleft and the postsynaptic cleft. Those things will separate one neuron from the other. Neurotransmitters are sent from the pre to the post. And there's a table on page 739 that you guys need to know. You need to be able to list out. There's 1, 2, 3, 4, 5, 6, 7, 8. There's 9 of those. You need to know 4 neurotransmitters. You need to know their location and 1 function for your test. So 4 of those, 1 location, 1 function. That will be a short answer on your test. And then figure 38.4 on page 737. All right, well on that one, you guys are going to have to understand how an action potential works. So I kind of already just went through that, talking about the sodium, the potassium pump, and the neurotransmitters. One thing to add in there that you guys need to know, neurotransmitters are released from vesicles at the end of the axon, and when those neurotransmitters are released, they open up the ion gate. So they open up the sodium gate for sodium to flow in to send the message from the pre to the post. Okay, so add that in, but that's basically what we just talked about. Now you'll see on your PowerPoint, there's a couple of YouTube videos that I found for you guys to watch, but you might find some different ones that you like better. Now on page 740, okay, page 740, it says read and summarize the closer look at synaptic transmission. So with this, they talk about action potentials reaching the presynaptic terminal. The positive charge inside the terminal opens a new set of voltage-gated ion channels, which are selectively permeable to calcium. The calcium concentration, the concentration of calcium outside the terminal is about 10,000 times higher than the concentration inside. Therefore, calcium enters the terminal, where it activates a series of proteins that cause vesicle-containing neurotransmitters to fuse with the presynaptic membrane and release their transmitter into the synaptic cleft. Now that's what I was just telling you guys. You got your pre and your post, and once you get that action potential down to the end of that axon, the neurotransmitters are released from vesicles. So the transmitters diffuse across the cleft and bind to receptor proteins on the postsynaptic cell. This typically has one of two effects. In some synapses, the result of transmitter receptor binding is similar to what happens when a peptide hormone binds to its receptor. Intracellular messengers are made, synthesized, and the metabolism of the postsynaptic cell changes. At most synapses, however, the receptor proteins are linked to ion channels, and that's what was important for us and what we saw. Those ion channels open up. So at most synapses, they're linked to the ion channels, and neurotransmitter binding opens the channels, like the sodium channel. If the channels are permeable to sodium, then sodium diffuses down its concentration gradient into the postsynaptic neuron, making the cell less negative, in turn more positive. If the postsynaptic neuron becomes sufficiently less negative, it reaches threshold and produces an action potential. Because they excite the postsynaptic cell, such voltage changes are called excitatory postsynaptic potentials. If the channels are permeable to potassium, then potassium diffuses out of the cell, making it more negative, making the cell more negative, and that inhibits the production of action potentials in the postsynaptic cell. So this voltage change is called an inhibitory postsynaptic potential. So a way to make action potentials happen is to let sodium flow in. A way to stop them from happening is let potassium flow out. So you guys need to know that for your test on that page. Now, so we've gotten to, we did inhibitory, we did excitatory. Neurotransmitter action is usually very brief, so it happens very quickly. Those neurotransmitters in the synaptic cleft are usually either degraded or taken back up by the postsynaptic cell. Another bold word in your book is integration on page 741. So where it says summation of postsynaptic potentials determines the activity of a neuron on page 740. Most postsynaptic potentials are really small. They rapidly fade away, but they travel far enough to reach the cell body. There they determine whether an action potential will be produced. So how does that happen? The dendrites and cell body of a single neuron often receive EPSs, right, EPSCs and IPSCs from synaptic terminals of thousands of presynaptic neurons. The voltages of all the PSCs that reach the postsynaptic cell body at about the same time are added up, a process called integration. If the excitatory and inhibitory postsynaptic potentials, when added together, raise the electrical potential inside the neuron above threshold, then an action potential is reached. So what this is basically saying, if the information from the presynaptic neuron is not great enough when it reaches the cell body of the next neuron. If it's not great enough for threshold, then the action potential won't continue. But if it is great enough, then it will continue. So just because the message went from one neuron to the next doesn't mean the cell body of the next neuron is actually going to create threshold and then create an action potential. So on page 741, we're going to stop with this test at 38.3. So 38.3 will be on the next test. Now what I'm looking at now is your test and some things you need to know. You need to know the part of the neuron that receives information. You need to know the part of the neuron where electrical signals are transmitted to. You need to know the path a nerve impulse travels within a single neuron. You need to know the place where a neuron communicates, what it's called. You need to know the function and what an axon is. You need to know about the sodium-potassium pump. You need to know what channels are open and closed at resting potential. You need to know when an action potential reaches a portion of the membrane, what opens, what channels open. You need to know the voltage of resting potential of a neuron. You need to know the function of myelin, the function of the sodium-potassium pump. You need to know what happens when a neuron reaches threshold. You need to know what causes action potentials and resting potentials to occur. You need to know the main neurotransmitter that is important with the nervous system and skeletal muscles. You need to know which neurotransmitter is important in forming memory, which neurotransmitter is linked to mood and sleep. You need to know where neurotransmitters are released. You need to know what nerves are. You need to know the function of myelin. You need to know the part of the neuron that contains the nucleus. And then really the difference between action and resting potential. The rest of your test will be short answer questions. We're going over all those in class. So make sure if you have any questions, you ask me in class, or you email me or send me a text message.